Hello and welcome back, everybody. In today's lecture, we're going to be continuing our journey down the biological links between the mind and the nervous system. But what we're going to be doing in this particular lecture is starting to kind of create that bridge between people studying the mind through biological processes with those who study the mind through more cognitive or mental processes. The way we're going to do this is talk about two very intertwined topics that have actually been a source of interest since psychology was formed over 140 years ago at this point. It's this wonderful area of what we call sensation and perception. And to understand how it is we make sense of all the stuff in our environment, we actually have to appreciate that it's sort of a two-step process. The first process, it requires us to take all of the different types of information that are present in our environment and find a way to convert them into something that our brain can then decode and kind of represent mentally. That process of converting energy in a whole bunch of different forms into some type of neural signal is a process of what we call sensation. And it's a pretty heavily biologically based area of interest. The other area, perception, is where we start getting into the mental side of things, because in perception, what we're doing is actually taking that code, that signal that's been sent to the brain, and finding a way to represent it in something that we can understand mentally, and in doing so, which we'll be talking about in future classes, and react to that information that we are perceiving. We will be getting to the perceptual processes in our next class. It's a very important step to take because it will lead us into a new branch of psychology called cognitive psychology. But for this lecture, we're going to focus our intentions on the first step, our attention on the first step, that process of what we again call sensation. So how does sensation specifically work? To understand this process of sensation, we first have to appreciate that we actually have a multitude of different what we call sensory cells throughout our body that are responsible for all the different senses that we have. You might have heard of the 5 different senses. There are many psychologists that argue that we actually have way more than that if we look at all the different aspects of touch that are there. We'll talk about that when we get into the touch lecture a little bit later. But know that even if we're breaking down our senses into just the 5 different types, we actually have a number of very specialized cells that get this sensation conversation, we'll call it, started. When we look at hearing, what we see is we have very specialized tiny hair follicle cells that are actually moved through the vibrations of information coming through the air that actually activate another, well, actually, it's a little bit more complex than that. We actually have air pressure changing, which fluctuates a tiny flap of skin called the eardrum. That's that kind of border between the inner ear and the outer ear that then triggers movement of 3 different bones, which then connects to kind of a liquid-like substance that's contained within something that sort of looks like a snail, and on a strip within that snail-like structure are these hair follicles that actually bend and move as there's changes in the air pressure around us that we then eventually find a way to represent as sound. It's actually a couple weeks that you'll dedicate if you take a sensation and perception class to how you're able to convert the changes in air pressure into some idea of sound. But just understanding how cool this first step is, I think, is worth covering in a Psych 1 class. Same thing with touch. In fact, I'd like to spend an entire section talking about it when we can, because touch is a very multifaceted system that allows us to pick up on not only things like changes in pressure and temperature, but subtle changes that we don't necessarily think of. We're going to talk a little bit later about stuff like proprioception and interoception, 2 types of internal-based touch that actually involve sensory cells that are found within our body, not necessarily on the surface of our skin. And all of these sensory cells find ways to collect certain elements of the information around us, the energies around us, So they can send that information to our brain. One of the areas where it goes to is an area we talked about earlier, the primary somatosensory cortex. So we can then disperse that information to other areas of our brain and start to make sense of it. That's again, though, the perceptual side of things, and just understanding the coolness of the skin and all the things inside our body that help us with touch is what we're looking at when we talk about sensation. We also have really cool sensory cells on our tongue and our nose that are actually sensitive to different molecule combinations. And their ability to pick up on a wide array of different molecule combinations and distinguish between subtle differences within them is still something that we're tinkering with and trying to understand today. But we do know that you have specific cells, structures that are designed to essentially grab onto the molecules passing through your nose or passing through your mouth, and actually find a way to take that information, or not passing through your mouth, passing along your tongue, and take that information and create some type of neural signal that our brain then can start to try to decode. And all of these different senses are worth talking about, but usually when people talk about the sensation and perception steps, what most people do in, say, an intro class like this is focus their attention on our very commonly studied sensation, one of the ones that's actually the most popular over time, and that is our sense of vision. And you might ask, first off, when we talk about vision, what does it actually entail? Right? We can understand the idea of molecules latching onto things and then us breaking down that code, even if we don't know how we break down that code when we talk about it in touch and smell. And we can sort of understand how movement of certain follicles in our ear or on our skin could then create this cascade of neural communication that eventually gets to our brain. But what the heck are we doing when we see? What is it that we're actually converting into some type of neural signal? To understand what we're converting, we first have to understand some basic principles, properties of light. Now, I know there are probably many physics majors out there and biology majors and even chemistry majors, if we want to break it down even further, that have their own definitions of what light is. It's a very multi-pronged thing that you can approach from a number of different angles, but in the psych class, we talk about light as really a byproduct of an electromagnetic source. Right? These sources essentially project these energy waves, these energy pulses that in our visual system are represented as light. And what we have in the back of our eyes are very specialized cells that can actually convert this electromagnetic energy coming from these light sources and create some type of neural signal out of it that differentiates between the two main components of that electromagnetic energy, both the amplitude and the wavelength of that information. Now, some of you might be asking, wait, what are sources of electromagnetic energy? You've probably encountered many of them even in the last couple minutes. The sun is our primary source of electromagnetic energy if we look at the history of human beings, but there's lots of other sources of electromagnetic energy out there. Some species actually produce their own electromagnetic energy, like fireflies and other animals, but usually when we're talking about electromagnetic energy, it's something that's emitting massive amounts of energy that we're able to detect some of because of the specific types of energy that our eyes are sensitive to. And again, we can, in our eyes, differentiate that energy into two different categories. Now, the first component to light that we can talk about in this class is something called the wavelength, or the frequency of the light that's hitting our eyes. I guess you would call it the frequency of the electromagnetic pulses that are hitting our eyes. In a textbook, in most conversations, even the words wavelength, you get this sense that wavelength is all about kind of the magnitude of the waves that the light is hitting us in, or kind of how frequently the waves are hitting our eyes. That's actually a misnomer. If you want to think of wavelength in kind of a logical way, think of electromagnetic energy is this kind of constant energy that's pulsating and hitting our eyes at very frequent rates. And essentially, some of the light hitting our eyes is hitting us at a very fast rate, while other times the light hitting our eyes is hitting us at a very slow rate. But even talking about it in that particular means isn't really doing this electromagnetic energy wavelength true justice. And that's because when we're talking about how we measure the distance between wavelengths of electromagnetic energy that are represented in different lights, what we see is that we have to measure electromagnetic pulses in this very microscopic distance that we call nanometers. So all light travels essentially at the same speed, but when light gets to our eyes, it's either hitting us at a very, very, very fast, rapid speed or a slightly slower speed. The higher the wavelength, essentially the slower the time, or actually the longer the time it is between pulses hitting us. But again, the long distance between them is in nanometers, not something that you'd ever really be able to measure with, say, a measurement tape or a measurement stick. As we move down in wavelength, that means that the light is actually more compact. And it's really important to note, as you can see in this slide, that in actuality, our eyes, even though they're great at picking up on certain wavelengths and differentiating between them, are only picking up on a small subsample of the different wavelengths of electromagnetic pulses that are around us at any given time. Essentially, a lot of electromagnetic sources are out there, and they're constantly emitting this electromagnetic energy. It's just our eyes aren't sensitive to the rhythms, we could say, of the light that's coming out from them. So we don't even recognize them, even though they can be just as impactful as the light that we can actually see. Now, the other thing that we're able to differentiate between when looking at how we can break down electromagnetic energy is the amplitude of the light waves that are hitting us. Even though it's kind of difficult to, again, give a good representation of what amplitude looks like, it's probably important to note that most of your textbooks usually represent amplitude of electromagnetic energy and the differentiations between it very poorly. You'll often see in textbooks a picture like the one in the lower right-hand corner where small amplitude light is represented by a very low slithering snake, and great amplitude light or high amplitude light is represented by this extremely large kind of sliding snake that goes across the screen. It suggests that somehow the height of the waves can vary from electromagnetic source to electromagnetic source. It's not what we're looking at here when we talk about variations in amplitude. When we're talking about variations in amplitude of light, we're talking about how kind of compact the light is and how robust electromagnetic pulses are when they hit our eyes. When something's very low in amplitude, essentially it's not very impactful. There's not a lot of the electromagnetic pulse hitting us at whatever rate that pulse is hitting us at. And when it's represented in the mind, what's represented through is just a very dim light. High amplitude light, as you can probably guess now based on the low amplitude discussion, is one that is represented by really bright lights, and it's usually very compact, very powerful light that's hitting us at, again, whatever frequency it's hitting us at. And when we see something as bright or dim, it can be a whole variety of different colors, but the actual brightness of it is something that we're making sense of due to the amplitude of those electromagnetic pulses that are hitting our eyes. So how are our eyes seeing then? What allows them to convert these elements of the electromagnetic energy coming from these sources into something that our brains can make sense of? Well, what allows us to do this is actually a collection of specialized cells that are found not in the front of our eyes, but the back of our eyes. Light essentially comes from these electromagnetic sources, travels through the eyes, reaches the back of them, and then they start this cascade of neural information that eventually works its way to the back of our brains so we can start to process it mentally. In some classes, you might start by focusing on those cells in the back of the eyes and how they're able to convert that energy into something our brain can eventually make sense of. But in this class, I'd like to start a little bit further forward. I like to start with the different structures of the eyes that light is passing through, because they're pretty critical players in how we're able to decode what it is that's around us. And there's nuances to them that can allow us to really appreciate the sophistication of sensation. And also, it will allow us to bridge the gap between sensation and perception much more easily in our next class. So let's start there. Let's break down the eye, kind of dissect the different components to it, so we can really understand this process of vision a little bit better. Now, when we talk about the structures of the eye and how information goes from the environment down to the back of the eyes, I always like to start in the exact same place. And it's actually something that, even though we think of it as a structure, is not actually physically a structure at all. It's something that we call the pupil. And for all of us, our pupils look black. And that's sort of a tell as to what the pupil actually is. Because even though it sort of looks like a structure, what it really is just a gaping hole. A hole that allows electromagnetic energy from the outside environment to eventually funnel through our eyes and reach the back, where we do have those cells that can convert that electromagnetic energy into some type of neural signal. It's black because essentially we don't have light or electromagnetic energy coming from the back of our eyes. And the electromagnetic energy that goes in doesn't get to bounce around our eyes. It gets absorbed. So all of us have pupils that look exactly the exact same way. Well, exactly the same way. Around the pupil, though, is a really interesting combination of muscles that make up what we call the iris. Now, what's neat with the iris is that even though all of us have the same colored pupils, there's a wide range of different irises that are out there. Some people have brown ones, others have gray ones, others have blue ones. It comes in a variety of different color combinations, and that's partially because there's lots of genetic combinations that are linked to our eye color, which seems kind of weird. And what's even more unusual for researchers, we don't really have the ability to trace differences in eye colors to a lot of differences in color capabilities or even differences in how people see things. But nonetheless, regardless of the color of our iris, what we see is that these structures are really responsible for one thing and one thing in particular. They allow the light passing through the eyes to vary. So when it's really dark, what the iris will do is tighten, and in doing so, kind of contrast, get very small and thin, and by doing that, it'll make the pupil, that gaping hole, get larger so more electromagnetic energy can be let in and reach the back of our eyes. When it's very bright out, well, the iris responds in kind and it gets larger, and that prevents a lot of electromagnetic energy from reaching the back of our eyes, and thus saves us from having our brain and other cortical regions sort of overloaded with information and stimulation. This is the primary function of the iris, but what's kind of neat, and the way we can tie this to psychology, is the fact that there are some things that we've discovered over the years that sort of override this function of the iris. A classic example of this is actually something that we see in our social psychology research, where we can actually gauge how interested or invested somebody is in something, not by asking them how interested they are, but just by zooming into their eyes and looking for how their iris is responding to the situation. What we know is that if somebody finds something engaging, or at least something that's activating and getting their nervous systems to kind of go into overdrive, this iris will actually restrict, and it will allow more light in regardless of how bright it is outside. If somebody's fairly bored or they're very subdued, the iris will essentially get tight and not let a lot of information, a lot of light in. And that's kind of a really interesting effect that, again, you can explore in a lot of different ways. My favorite example is if you say, go out on a date with somebody and you want to know how interested they are, stop worrying about whether or not they're touching their hair or their face or who they're talking with. And just look at their eyes. And if it's a fairly normal amount of light in the room and you're seeing two giant orbs staring back at you, things are probably going pretty well. If you're staring at two tiny little dots, you've probably boreded them to death, and you don't really need to expect a second date at that point. Is also just one of the many different ways that the iris can be impacted, though. Another one that you'll learn about in a drugs in the brain class somewhere down the road if you ever take it, is that the iris can be overridden by a lot of different types of drugs. Essentially, there's lots of chemicals that can slow the functioning of the loop between our brain and the iris, and in doing so, it can prevent the iris from responding to massive amounts of light as quickly as it should. This might explain why, if you've ever had a friend, or maybe you yourself have been pulled over in the middle of the night by a police officer, why they immediately flash those lights in your eyes. What they've been trained to do when doing that is testing whether or not you have something in your system that's essentially overriding the automatic function of this structure, the eyes. And it means even if you tell that police officer that you swear you've had no drinks, nothing else that's been ingested, they might not believe you so much. It's really a byproduct of what your biology is doing that kind of determines how your iris is going to respond. And you can't, unfortunately, just tell it to respond the way you'd like. There are other things as well in mental health and types of antipsychotic medications and antidepressants that are out there that also have the same effect. So there is a need to not immediately judge exactly what's going on with somebody just by looking at how their eyes reacting. But all of these really interesting tidbits about how the iris works and then also how we can override it really highlights kind of that wonderful merger between biology and psychology that's out there. And this is also true for another pair of structures that are really around the pupil and the iris. The first structure, which I want to cover just because it helps me lead into the other one a little bit better later on, is something called the cornea. Now, the primary purpose of the cornea is to help protect the eyes. It's this rigid structure that's on the surface of the eyes, right near the front, and this structure has the consistency of essentially your fingernails. It's opaque-ish, not perfectly opaque, but fairly opaque. And that helps prevent a lot of potential things that could damage or hurt your eyes from ever getting to that point. The cornea also has the ability to, and this is kind of neat, bend the light that's coming in from the outside environment. You might not think of that as something that's that critical, but what it's doing in its attempts to bend the light is kind of minimizing the amount of space in the back of your eyes that's needed for you to be able to see in your entire visual field. And that's got a lot of utility. It saves us a lot of energy, a lot less need for integration, and essentially we can be more efficient, which is very important when it comes to how quickly we need to sometimes react to things that are in our visual field. So the cornea serves a very important purpose, even if it's kind of overshadowed by another part that's right behind it called the lens when it comes to bending the light. But before we get to that structure, you want to go back to the cornea for one more second and identify that just like there was issues with the iris and kind of the mental world, there's also issues with the cornea. So what many psychologists have discovered over the years, well, biologists really have discovered over the years, is that the curvature of the cornea and the thickness of it, as well as some of the imperfections that are persistent in almost everybody's cornea by the time they become adults, suggests that most of us should see the world in a very weird way. We should essentially be seeing the world in this flattened, condensed version with lots of imperfections based on all the scratches and random things that have occurred to the cornea over the years of our lives. But almost none of us see the world in this manner. None of us catch these imperfections, and in fact, most of us probably see the world as perfectly crystal clear. How does that happen? Of course, obviously, I know there's some of you right now thinking, oh, actually, I need glasses. My cornea is not working great. That's not what we're necessarily talking about here. We're talking about the inability to focus, but the clarity of the images that we're seeing tend to be universally seen by all of us. How can we do that?
Well, to answer this question, I want to now go to the lens, which again is another structure that's very critical for bending light. But the lens doesn't just bend light, so we don't need a lot of space in the back of our eyes to process information. What the lens is essential for is being able to bend light in a way that allows us to focus on information at different distances. So when something is, say, just a few inches in front of your face, well, a few feet in front of your face to be fair, our lens has to bend a lot, but in doing so, it can allow us to focus on that thing that's right in front of us. If something moves further away, our lens quickly has to bend, and in doing so, that thing that's further away tends to come into focus. Think of it like a lens of a camera, where if you're not focused on the right thing, everything's blurry. Now, some of us might be questioning, one, are we actually doing that? Right? Are we actually adjusting our lens all the time? Because we almost never see anything in our visual field as blurry. And the reason for this is because the lens is really, really good. If we were in class, we'd do this really fun activity where I have you point your thumb at me, putting your thumb just a little bit below my face, and then focus your attention on your thumb. And what you'll see when you focus your attention on your thumb and just sort of catch me in your periphery is that I become very blurry when you're focusing on your thumb. And when you focus on me, while your thumb is still sticking out, your thumb starts to become blurry. Going back and forth between me and your thumb is exceptionally easy. And that's essentially what we're doing at any given time when we're focusing on things at different depths. We're quickly bending, tweaking our lens, so the thing we're looking at almost always looks clear, and we don't even really notice the stuff that we're not looking at as being fuzzy or unclear. The other thing you might be saying is, for me, things aren't clear at different distances. You might be farsighted or nearsighted. I promise you, this is almost never the fault of the lens. The lens is essentially doing the best it can with what it has. The curvature of your eyes can be partially the curvature of your cornea, but other things as well are usually the source of visual acuity issues. There's also issues with musculature in our eyes and whether or not we can focus on things clearly, especially as we get older. But know that for all of us, well, most of us, the lens is doing the best it can. But what happens as a result of the lens is kind of interesting. There's numerous people who have looked at the eye and the structure in the lens and come to the conclusion that the way light is bent through the lens, we know that for all of us, a really interesting consequence is produced as a byproduct of that light bending. That light, that information that's coming from the outside environment, appears to be, if all of our calculations are correct, actually flipped by the time that information gets to the back of our eyes. So up is down and right is left. When we're looking at kind of the corresponding cells in the back of our eyes picking up on this information, which of course leads to the odd, weird question of, do we see the world as upside down? I'm guessing most of you, when you hear that, pause, wonder what the heck I'm asking, and then come up with the very straightforward, obvious answer of, no, of course we don't see the world as upside down and flipped, right? That's just not what we're seeing when we're processing the world. Just like, as I talked about with the cornea, we don't see the world as sort of milky and bent and distorted like we're looking through a dirty periscope. But how is this possible then? What allows us to make all of these course corrections so we can persist in thinking that what we're seeing is relatively clear, even with all of these issues? Now, to understand this process, we had to go down a number of different avenues. Now, historically, what we were able to do to kind of test how the brain was able to flip things, even before we could look at the brain itself, was to develop tools that essentially mimicked the effects that we knew the lens were creating. There was really cool pairs of glasses that researchers designed years ago, many years ago, over a century ago. They would put on to people, and when people put on these glasses, the curvature of the glass in these glasses actually cause the images hitting our eyes to be flipped, to be switched around. And these individuals would then be prompted to do basic tasks like play pin the tail on the donkey or catch or try to follow the movements, shadow the movements of a researcher. And as you could probably expect, the people performing these tasks when they couldn't see the world right side up was pretty abysmal. Those people did not do exceptionally well on these tasks. And this, of course, led to the question of, well, how? How could we do better? What would be the catalyst for us to perform better? So researchers decided to keep these glasses on these individuals for a while and see if they could perform better. What they started to find in numerous studies was that as they practiced, their performance kept getting better and better and better. And then, surprisingly, another effect started to emerge as they got better and better. Despite the fact that the world was still upside down and reversed, many people started to report that the world started to kind of reorient itself. Eventually, they all reported, as they progressed through a couple minutes or hours of this, depending upon who they were, most of them reported starting to see the world as right side up again. And this is really amazing because you have to remember, they still had these glasses on. The world was still flipped and twisted, but they didn't see it that way anymore. Somehow their mind, their brain, had rewritten the entire mapping, the formula that we were using to understand where things were in a very short period of time. And this led to the wonderful question of, well, what happens if we take the glasses off? What many of the researchers found when they did this to people was that the effects were pretty darn robust. And it's one thing to put on a pair of glasses and have the world flipped upside down on you. It's a whole different thing if you're having a pair of glasses that are supposed to be flipping the world upside down taken off, and suddenly now you see the world is upside down, which is what many people reported. Now, there were people that were pretty distraught by this experience when it was taken off, but for those of you worrying, know that people's vision resumed very quickly when the glasses came off, but there was a lag. There was a delay for the mind to kind of need to catch up and recalculate things out. And for a long period of time, we just said, look, this is something that's going on in the mind. How we can adjust to the cornea, how we can adjust to the lens is just the mind reconfiguring things. As neuroscience started to become more popular, what people were able to do is actually detect certain regions of the face, of the brain, that seem to be responsible for us kind of reorienting ourselves when things start to tweak. Sometimes these responses can be very rapid. Other times they take a little longer, but essentially by having them there, we don't catch the vast majority of imperfections that are constantly around in our visual fields. A favorite example of this that I have is when we have maybe an eyelash that gets bent or something on our nose that causes it not to be the same shape that we're accustomed to. We'll notice that eyelash. We'll notice that new bending in the nose for quite some time. And then eventually, it'll just magically go away. It's not that those things are gone per se. It's just that our brain has overwritten that information and now allowed us to see things as they were before those imperfections came into play. Now, after we get through the lens, we get to an area that's called the vitreous body. And it's called this because it's filled with another substance that we call the vitreous humor. And it's actually one of two types of humors that we have in our eyes. The first is called the aqueous humor, and both are kind of like a jelly-like substance that have the consistency of somewhere between, I would say, pudding and jello. And these substances help keep the eye inflated, help keep the structures intact, and sort of are protective of the rest of the structures of the eyes that are significantly more fragile. The important thing with the vitreous and aqueous humor is that we don't drain it from the eyes. It's very difficult, if not impossible, to recreate the structure of the eye if we do puncture it. And usually that vitreous body is permanently damaged if we do happen to rupture it, which is another reason why that cornea that we talked about before is so unbelievably important. The protective abilities of it cannot be overlooked, even though it does serve the purpose again of also helping to narrow the amount of light that's hitting the back of our eyes. As the light passes through that vitreous humor, it eventually works its way to the back of the eyes and hits a large area that's called the retina. Now, the term retina is usually used for the entirety of the area in the back of our eyes that have those receptor cells that start converting the electromagnetic energy into neural signals. There is, however, one specific area of the retina that extra attention is paid to, and this is a structure, not necessarily structure, but an area that we call the fovea. And to understand the importance of the fovea, a very narrow band along the retina, which is directly in the back of these eyes, we have to understand that when we talk about cells converting electromagnetic energy into some type of neural signal, they come in different forms. There's two different types of cells that we have that do this conversion process. The first type of cell is called a rod. And they're called rods very aptly because if you look at them under a microscope, they look like tiny rods sticking out of your eyes. Usually we tend to link rods to light adaptation and not light adaptation, but dim light processing and the ability to kind of detect the presence of light, even magnitudes of light. So if we're going back to the differentiations of different components of electromagnetic energy, you could say these ones are more sensitive to just sort of changes in the amplitude of the light, not necessarily changes in the other component of light, and that's the frequency of the light. And that's because we have other cells that are more attentive to this, and we call these cells cones. Again, they're aptly named cones because they look like, under a microscope, the traffic cones that you would see on the highway or somewhere around there. If we're looking for where these cones are located, this is when we go back to that fovea area that we just spoke about. The vast majority of our cones are concentrated in the fovea, and this allows for us to process the wide array of colors that we can see much better. If you've never noticed that whenever you look at something, try to understand its color, you have to look at it directly on, you maybe have missed the fact that this is a kind of necessity for our color vision, and it's a byproduct of those multitudes of cones that we have in our fovea. If we tried to understand the color of something by just looking at it in our periphery, where it's primarily rods, what we tend to do is be able to sort of make the shape out of that object that's in our periphery, but not be able to pick up on the fine details of the color or even the kind of small elements of that thing that we're trying to detect. Now, that doesn't mean that our rods are useless. They do have a lot of utility in us being able to detect the presence of things, which can be essential in our ability to kind of navigate in our everyday world. But if we are looking at fine-tuned processing, it's usually reserved for cones and all of the really amazing things that they can do. Now, some of you might be wondering now, well, if cones are responsible for picking up on different frequencies, how do they do it? That's going to be something we're actually going to discuss in our next class. I'm not going to give away too much, but do know that we will come back to this at that point. There is one last spot of the eye I do want to discuss before we move to kind of the transition to the brain. And it's actually kind of like the pupil, a spot that really isn't a spot. It's actually sort of a path out. It's a hole where there isn't the presence of any rods or cones in the retina, because we need to find a way to get the neural signal that those rods and cones are generating out of the eye and straight to the brain. Well, eventually to the brain, I guess. We'll talk about that in the next slide. But there's a byproduct of this path out. It's something that we call the blind spot. much like our inability to pick up on the fact that our lens flips the image that's hitting the back of our eyes, much like we're incapable of being able to detect the imperfections in our eye structures because our brain just fills in the gaps. Well, the same thing happens for the blind spot. It's almost impossible to detect that both of our eyes actually have a blind spot in them. And that's because our mind has this apt ability to fill in the blanks through previous experiences, memories, and kind of expectations of what should be present in that blind spot. And that second eye does really help with not having to worry about the blind spot most of the time. That doesn't mean we can't detect the presence of the blind spot if we create the correct scenario. Your textbook, I think still in this edition, should have an activity that asks you to cover one eye while looking over the top of an image of, I believe it's a lion tamer with a lion. You're supposed to center your eye over the lion tamer and just make sure that the lion is in the far periphery of you, regardless of which eye you're using. So if it's your right eye, it's going to be on the far right side. If it's your left eye that you're using for the activity, you would put it on the far left side. And then you're going to back slowly away from the lion tamer, and eventually what will happen is you will just see not the lion tamer with the circle and the lion, but the lion tamer standing there alone with a complete circle. And that's the point where you've hit the blind spot. The lion in particular has hit the blind spot and just disappears, but because your brain doesn't like things just disappearing, it fills in the blanks regardless of whether or not you want it to, and it creates this complete circle. We'll again look at this a little bit more closely when we get into our next lecture on perception. But I just wanted to kind of give you an example of another component of the eye and how it's kind of quirky and related to the mind as well. After that information does get to this, again, what we call optic nerve, that information travels down and meets this juncture that we call the optic chiasm. It's the area where the information from our left and right eye meets and gets filtered in sort of peculiar way. To understand this filtering system, we have to go back to something that we spoke about in our previous class, this idea of something called contralateral communication. In that class, we discussed how large portions of your body are controlled by certain hemispheres of the brain. In particular, your left side of your body, both in terms of touch and movement, is often dictated by the right side of your brain. And in converse, your right side of your body is controlled or kind of detected by the left side of your brain. That's how contralateral communication works for touch. For vision, the story is a little bit more complex. It's not that your left eye is solely dedicated to the right side of your brain and your right eye is solely dedicated to the left side of your brain, but contralateral communication does occur for vision. It's just for vision, we talk about these things called visual fields. To envision visual fields, what you need to do is just look straight forward and sort of imagine a line extending from your nose straight out, and think that everything on your left side, as you're looking straight out, if you look to the left, you mess with your visual field, but if you continue looking straight out, everything to your left is part of your left visual field. Everything to your right is a part of your right visual field. Well, these visual fields are the things that are related to contralateral communication when it comes to vision. So your left visual field, just like with their bodies, is controlled by the right side of your brain. And your right visual field is controlled, or at least processed, I guess, by the left side of your brain. And this optic chiasm, the structure that we were just referencing, is the part that does that filtering. And you can actually see, if you look closely at this picture, that there's information that's in black lines and red lines coming from both eyes, and that information gets sorted and splits to the different sides of the brain. If you look even more closely at this, you'll notice all the information hitting the right side of our eyes is going to the right side of our brain, and the left side of our eyes is going to the left side of our brain. This is not an error. I know many of you are thinking they must have screwed up. That's not what they did. In fact, this is exactly what we see. You have to remember, when light passes through our eyes, it passes through our eyes in a straight line. Therefore, the information on the left side of our visual field passes through our eyes, and because it doesn't do a weird curving hook, it continues through our eyes and goes to the opposite side of our eyes. So left visual field information hits the right side of both our left and right eyes. Right visual field information, because it does not curve or do anything weird once it hits the eyes, goes directly to the left side of both our left and right eyes. I know this can be confusing. It might take a while to kind of process what this means, but that's exactly what's going on when it comes to contralateral communication with vision. And the structure that allows for it to occur is that optic chiasm. Some of you might be thinking, why do we have this complexity? What's the point of it? Well, again, we're not sure why contralateral communication happens, but that partitioning of the information, or visual information, can serve us really well. Because what it allows us to do is coordinate between the different hemispheres, all those different sections, very quickly. So if we see something in our right periphery, we can quickly react to it with parts of the right side of our body without sometimes even thinking about it. And that's because of the fast path between the different structures of our left hemisphere of our brain that are all processing that visual information and motor systems on our right side of our bodies. Another thing that helps with this is a structure that's very critical in almost all of our senses called the thalamus. The thalamus is sort of a gatekeeper slash operator for all of the different senses that are trying to find their way up to the brain. We have almost every single sensory organ. That's our skin, our eyes. The only exception to this is our nose. But outside of that, every sensory organ that we have sends a message first to the thalamus before it goes to what we call the projection areas, the areas of the brain that are going to decode and start to break down that information. And this stop off at the thalamus allows us to do a number of things. The first thing it does is it allows us to filter stuff out. If we didn't have this thalamus, our brain would constantly be bombarded by all of the information that we're being presented at any given moment. We might not appreciate it, but right now, as you're watching this video, you've got many sounds that are hitting your ears beyond just the sounds of my voice. You have all of these different things that you're seeing, but hopefully there's only a certain number of things that you're visually focusing on right now. You have all of these different tactile sensations that are hitting you. You might not have noticed the clothes on your skin for quite some time, but they're still activating different cells trying to get messages to your brain. Well, I wouldn't say that clothes are trying to get messages to your brain, but there are signals being sent to your brain, and your thalamus is just filtering a large number of them out because, well, you don't really need to process how your clothes feel on you or your chair feels unless there's something critical that you need to pay attention to. So again, the biggest function of the thalamus, I would say, is the filtering function. But another thing that the thalamus is responsible for is being able to quickly coordinate actions through our body that don't require lots of higher cortical thinking. So if, say, somebody were to toss a ball at you, well, the thalamus would be able to fast track that visual information coming in from primarily, I'm guessing, the rods in this case. So your brain could quickly then react to it without you even recognizing that you were reacting to it, causing this kind of cascade of information to communicate between the visual cortex and the motor cortex and have us have almost a reflexively response to that movement. This is very advantageous. It just is somewhat embarrassing sometimes when somebody throws, say, a light pillow at us and we just weren't paying attention and we freak out and immediately start violently shaking or jumping away from that very soft, not dangerous thing. it's a survival thing. It's very important for us to have those knee-jerk reactions, even if it also is followed by some embarrassing moments in our life. That really does highlight the multitude of things that this thalamus is capable of doing for us. But there is information, we're going back to the visual processing portion of it, that does get through the thalamus more often than not. And in fact, a large amount of visual information gets through that thalamus, and it works its way back to our occipital lobe, which is also called, oftentimes in vision research, the visual cortex, primarily because this is, again, where most of our visual processing occurs. And if we're looking at where this information tends to go, it usually goes to the very back of our brains first. When that information hits the back of our brains, this is when we start actually decoding that information. If you get into a neuroscience class, you'll learn about all these different vision areas that are responsible for different levels of processing of the visual information, and then paths that descend from this visual starting point that allow us to pick up on the whats and the wheres and all of the different components of that visual information that are really complex, but nonetheless we're able to break down. And I promise we will get to a lot of this breaking down portion in this class, not nearly as much as you would in, say, a neuroscience class or a sensation and perception class. But it's important to note here that when we start talking about the brain breaking down this information, we're getting to the next step on the way. We're getting to perception, which is where we're going to be going in our next class. So for today, we're going to close up. Sorry, I just noticed I went a little over time this time. Started kind of going off, I think, on the thalamus a little more than I expected, but hopefully you got a lot of good information there. We're going to close up for today. In the next class, we will be getting into the wonderful world of perception and explain some of the stuff that we discussed here. Maybe give you a little information about why you see those things as spinning, even though, remember, these are just slides, not videos. And yeah. Hopefully everything went well today, and I'll see you all very soon. Take care.